Hello everyone, welcome to today's video, and today we will be understanding inflammatory response to tissue injury. In this one, I'll try to explain how tissue injury is able to induce an inflammatory response in our body and how various molecules such as cytokines and various cells such as neutrophils, mast cells, they are involved in this tissue response or inflammatory response against the injury. And I'll also be talking about the connection between bacteria and the cells that are there in our immune system and how our cells are they are able to reduce or they're able to eliminate the microorganisms that are entering by the process of tissue injury. Okay, so let's get started. Before we start the video, I have a quick request to make. If you are new to the channel, then please support the channel. I'm creating lots of videos on various topics of biosciences, and if you show the support by just subscribing to the channel and also sharing the videos with your friends, that gives us huge motivation, and we should be able to create more videos on various topics. All right, with that note, let's get started. All right, everyone, so we are on the slide where we are going to create the entire illustration. I'll start with the background of my entire illustration, and then I'll start with the outermost layer that protects us from any injury or any damage. So in animals and in most living organisms, we have this protective layering that protects us from various infectious agents. So what happens is whenever there is any kind of injury or any kind of damage to this layer, which I'm representing by this illustration, as you can see over here, that a sharp object is penetrating the tissue, and there is this redness everywhere because the skin got damaged because of the sharp object. In this case, it could be a metal object, right? So it's getting penetrated inside the body, and because of that, what is happening is there is inflammatory response that is getting initiated. And because of that, also what will happen that a lot of microorganisms can also they can also enter into this environment. So let me label this first. So I have sharp object label for this, right? So this is the label that I'm going to use so that the understanding is clear. So this is the sharp object, and because of this, what is happening is a lot of tissue damage is happening in this region. So I can use this label to show the tissue damage in this region. So this is the sharp object, and I do want to move this one a little bit on that side because I'll be adding a lot of other things. So as you can see over here, we have redness, inflammation is there, tissue damage, which is the sign of tissue damage, and then we have the sharp object. Now, in addition to this one, we'll also have entry of microorganisms. In this case, let's assume that we have a lot of microbes, especially bacteria. They have entered into our system, and then these are some unwanted microbes, and we basically don't want them, right? So I can use another label to actually represent the cells also. Okay, so these are a group of cells, bacteria in this case, that are entering into our system because of the tissue damage. Okay. So there are a few steps I wanted to cover in this one. We'll start with step one. So in step one, what is happening? In step one, tissue injury and tissue damage is happening. When tissue injury happens because of a cut or because of a puncture of the skin, what will happen that mucosal barrier or any other barrier that is present is compromised. That will cause the exposure to environmental microbes, and that will lead to the induction of innate and the adaptive immune response. So in this one, what are the key events that are happening? Step one is the disruption of physical barriers such as skin or epithelial lining. And second one is necrosis of the cell at the site of injury, where you will have release of ATP molecules, uric acid molecules, and also heat shock proteins. They are going to be released that are, most of the time we'll call them as DAMPs, which is damage associated molecular patterns. So this is step one, where we are facing all these events all together, and our body is trying to respond against that, right? So second event in this case is the microbial entry into our body, and I've shown you that the cells in our body, for example, macrophages, dendritic cells, mast cells, they are going to respond against these bacteria, and they will detect the PRRs. They are known as pathogen pattern recognition receptors, such as toll-like receptors, such as knot-like receptors. They are there, and they detect pathogen-associated molecular patterns, in short known as PAMPs. So in our body, we have PRRs, and in the organisms, we have PAMPs. And because of this interaction, we should be able to identify what are the organisms that are entering in our body. So what are the key events that are happening in this case? Macrophages, dendritic cells, most of the time, they will recognize those PAMPs via using PRRs. And then PRRs, they will activate, and then important cytokines, they will be released. And those cytokines, they are TNF alpha, which is known as tumor necrosis factor alpha, and also I'll have interleukin 1 beta and interleukin 6. These are the most important cytokines that are released when there is activation of cells by using PRR. So that means the next step that we will be seeing in this case is the involvement of mast cells. So I think let's also create the mast cells right over here. And then mast cells along with the other factors like the cytokines that are released. And I have a nice illustration for that one. You can actually see, I'll show you the zoom in view. Look at this. So these are the small molecules, these are the cytokines, and we have discussed that TNF alpha, tumor necrosis factor alpha, interleukin 1 beta, and interleukin 6. These are the cytokines. They are going to be released, and then I can show this right over here. So what is happening because of this interaction with the cells, and also mast cells are there, macrophages are there, dendritic cells are there. They're going to cause the release of these inflammatory cytokines, and because of these cytokines, we'll have lot of activation that is going to happen in our body. And the next step will be there will be a response from our blood vessels also. So let me create that illustration also, so that everything is clear right over here. So what will happen that in this case, inflammation will also come from the tissue damage. So I'm using this arrow to show that the molecules, they are getting released by the tissue damage right over here. Molecules are getting released by the mast cells also. And then other cells that are activated by bacteria, they are also going to cause the activation of the small molecules, cytokines in this case. And then another component is will have the activation of the cells, especially the neutrophils, that are going to come and eat these organisms, phagocytize these organisms. So we need to show that. I'll use the blood vessel illustration. So this is what I've created right over here. So this is the blood vessel, which is very, very close to the site of the injury. And then in this case, what we have a lot of things over here. So response will be there in the form of cytokines from the blood vessels also. So I think that arrow also justifies that. So we'll have a response from the blood vessel also. But I need to show the internal part of this blood vessel. So I have used another illustration that will give you an idea that just assume that this is where we have cut down the outer part, and you can see the internal structure. So this is the internal structure. This is also the internal structure that you can see. So we'll have different types of cells over here. RBCs will be there. So let me move RBCs right over here so that we can see RBCs right over here, and more RBCs are inside this blood vessel. So it's now clear. We can see the RBCs inside. In addition to this, there are other cells, other factors, but I just wanted to show you the function of neutrophils. So I'll move this illustration, which is the illustration for the neutrophils. So as you can see over here, so these are the neutrophils. These are the neutrophils present in our body, and these are the cells. They are going to basically fight with the pathogenic organisms. So as you can see, this is our blood vessel, and this is also the part of inside of the blood vessel. And then now how these cells, they're going to move outside. So what will happen, a lot of molecules, they are getting released from this particular interactions, and they will cause change in the blood vessel. They will increase the permeability. So in this case, what will happen that cells, they will detect these molecules. The blood vessel cells, they will detect these molecules, and there will be increase in the permeability of these blood vessels so that the neutrophils, they can move outside. Right. So the next step here will be this, the movement of the cells, and this you have to be carefully visualize. What is happening in this case is cells, they are trying to move out from the blood vessel, and it's known as diapedesis. So in this process, what happens is cells, they are able to move outside the blood vessel, especially the neutrophils, because of the increased permeability, which is known as diapedesis. This movement where cells are moving outside, and these cells then they're going to work out on these bacteria, and they are trying to eat these bacteria and trying to phagocytize these. So I'll just use these two cells right over here, and what they are trying to do is they're trying to interact with the bacteria and trying to phagocytize these organisms that have entered in our body. So as you can see, what we have covered is we have covered entire inflammation process, role of RBCs, mast cells, and also the small molecules whenever there is a tissue damage in our body. Few of the additional things that I want to cover in this one is, let's discuss that step by step. So what will happen next is the activation of mast cells. So whenever there is this interaction, whenever the cytokines they are going to release, that will cause the mast cell to degranulate, and granules are there in the mast cell. They will be released. Histamine, heparin proteases, they will be released from the mast cell, and that will cause the vasodilation, increase the vascular permeability. That is what we have discussed. Right? What are the key events? Event number one is histamine release will cause the local vasodilation, and that will cause the increase of blood flow to the site of injury. Proteases and heparin that will promote the tissue remodeling, and also they will try to recruit more immune cells. You will have leukotrienes, prostaglandins, and these are the molecules that increase the vascular permeability. And the next step will be recruitment of the neutrophils to the site of injury. So what will happen in that case, we have also seen that by illustration, that because of these cytokines, which are TNF-alpha, IL-1 beta, IL-6, they will cause the neutrophil cells to actually come to the site of the injury. And using various molecules like E-selectin, P-selectin, and ICAM1, cells are able to move out of the blood vessel. So how that happens? So first step is migration, where neutrophils, they will adhere to the epithelium by using E-selectin. So that is the first step. Next one is rolling, where macrophages, they roll along the endothelium, and that will basically make the contact with the selectins. Next is adhesions and arrest. So chemokines such as CXCL8, which is also known as IL-8, will activate the neutrophils, which cause them to use various integrins, and because of that, the binding of LFA1 to ICAM1 will happen, and then diapedesis, which is the movement of neutrophils through the endothelial barrier, will happen. Next step is step 5, where neutrophils they will reach to the site of injury, and they will start phagocytizing, and then they will kill the organism by using reactive oxygen species, antimicrobial peptides such as defensins, and cause the destruction of the pathogen. In this one, key events are phagocytosis, which is eating up the bacteria. Second one is respiratory burst, where you have ROS, that are reactive oxygen species, getting generated. Next is release of enzymes that will cause the damage to the host. Right. So this is the overall process where inflammation, the process of inflammation is so controlled, so regulated that, every time if there is a tissue injury, this inflammatory response will make sure that no organism is going to enter inside the body. And if it enters inside the body, it should be able to kill by the various immune cells. And if there is any defect in those cellular processes, what will happen, that we'll get disease, we'll get infection. Right? So it's very, very important to understand all these processes, and a lot of research is also happening to understand these processes, so that we can find out various targets, we can enhance our immunity, and we can also target these pathogens efficiently. As we have understood, there are pattern recognition receptors, there are pathogen associated molecular patterns, and by understanding those things, we should be able to understand how the response of the immune system happens against these important pathogens. Right? So I hope the video was helpful for you to understand everything. I've tried to show you with illustration. I've tried to also explain the various processes that are happening with various important molecules that are getting released during the inflammation process. Okay, so if you like the video, if you like the work, then please do subscribe to the channel, and please do support us. I'll create another video, and if you have any suggestion, please feel free to put the topic that you want to understand. I'll try to make video on that one. All right, I'll meet you in the next video. Till then, take care, everyone.